Our speaker for today is Jacqueline Tyler Thomas, who works at the Emerging Technologies section at the Armed Force DNA Identification Lab. Tyler received her undergraduate degree in biology from the University of North Carolina at Chapel Hill in 2008 and a Master of Science in Forensic Sciences with a forensic biology concentration from Virginia Commonwealth University in 2011. And in 2011, she joined the Quality Control and Validation section of the Armed Force Medical Examiner Systems, Armed Forces DNA Identification Laboratory, or AFDIL. And in her 9 years in QC and validation, she's involved in projects from short tandem repeat kit validation to massively parallel sequencing for human identification, with roles ranging from completing lab work, managing projects, summarizing results, and to training of staff. And in 2020, she moved to the AFDIL Emerging Technologies section, where she primarily researches and tests new methods using historical cases in an effort to increase efficiency and success rates in processing of casework samples. We are really happy to have you here, Tyler. Today she'll be talking about recovering human mitogenome profiles from chemically treated US military unknowns, the intersection of forensics and ancient DNA.
Thank you for the introduction. I appreciate the ability to talk today about AFDIL. So as you mentioned, I'm going to be talking about recovering human mitogenome profiles from chemically treated US military unknowns. And this kind of falls at an intersection of the ancient DNA and forensic DNA communities. Standard disclaimers: these opinions are mine. They don't reflect the US government, the Department of Defense, the DHA, Armed Forces Medical Examiner System, and mention of commercial products isn't necessarily an endorsement or a recommendation. It's to specify protocols that we used for this research. So quickly, I'll start with background about the lab that I work for, including a brief history, its structure, and the types of samples that we encounter, routine casework. And then I'll talk specifically about our NGS or MPS process that we validated at AFDIL, our success rate with that protocol, and what we're doing to develop methods to work better on our more challenging samples. And that'll include single-stranded DNA library preparation kit testing. AFDIL was established back in the early 1990s when forensic DNA was still in its infancy. At that time, it was located just northwest of Washington, D.C. And 10 years ago, it was moved. The entire lab was moved to Dover Air Force Base in Delaware, which was a large undertaking, moving an entire lab without interruption of processing. But the reason it was moved to Dover Air Force Base is that Dover is the current nation's only port mortuary in the US, which means the remains of all deceased service members and other individuals supporting those overseas operations come to Dover. So the current day remains are the purview of AFMES, and AFDIL is a division within AFMES. We have about 120 DNA scientists and then additional support staff as well. And we are an accredited forensic laboratory, and we go through regular audits to maintain this accreditation. Currently, there are over 81,500 service members, primarily from the 4 conflicts listed on this slide, that are still missing. And about half of those are presumed lost at sea, such as ship losses or airplane crashes into the water. And then 75% of them are in the Indo-Pacific region. The DoD actually has an entire accounting command that is working towards this mission of identifying fallen service members. And that includes things such as accounts of battles, dental records, personal objects that are recovered, anthropologists are involved. And so DNA is a part of this accounting process. But because of this, the vast majority of AFDIL's casework is supporting this past accounting mission. Just quickly, our organization is split into a number of sections. We have a pathology investigation division that includes pathologists, of course, but also anthropologists, photographers, medical legal death investigators, mortuary affairs specialists. And this is for investigation into military deaths worldwide for the federal government. There's also a toxicology section that does routine drug testing for the military. They also support autopsies, drug testing for autopsies, sometimes criminal cases, things such as that. And then we also have support staff that includes IT, administrators, logistics, things of that nature. And then the DoD DNA operations itself is split into 2 groups. AFRISEER, which is where we house all of our reference samples for the current day mission, and also the Armed Forces DNA Identification Lab. And this again is split into multiple groups. We have our current day operations. We have a past accounting mission, which is our largest. We have quality management, which includes quality control and training. We have a family reference section that uses automation to process reference samples. And we also have the emerging technology section or research section. And that's a section that I'm a part of. This section, we have our hands in a lot of different functions within AFDIL. Perhaps one of the most interesting is optimizing and adapting DNA methods for the past accounting mission. And this includes use of historical cases, some of which are listed on the right in the orange box. And we use these historical cases because they can help simulate the range of quality and quantity that we see with our past accounting samples. They range from remains that are approximately 75 years old to as much as Viking era samples from that are about 1300 years old. Another portion of our work is that we are currently validating SNP capture and kinship analysis. Most of our past accounting samples, we don't have direct DNA references for these samples. So we use indirect references such as family members to do these IDs. And as time passes, the family members available for reference are more and more distant from the service member. SNP capture and kinship analysis will allow us to harness these reference samples. And it's also, as an NGS protocol, will be amenable to our degraded low quality samples that we need it to be. It will allow us to use both maternal and paternal references because with our current protocol that's non-SNP, we can only use maternal relatives, obviously, for a mitochondrial comparison or paternal relatives for Y chromosomal markers. And this will also help us to individualize samples more effectively than these two haploid directly inherited markers. So just to touch on our samples, when we consider all of our different sections at AFDIL, the samples range widely in quality and quantity. Our current day section typically is using fresh tissue samples or well-preserved bone samples. And a lot of times they can use traditional forensic methods for identification, which are STRs, short tandem repeats. These are autosomal markers that are well characterized for a forensic setting and can individualize people. And then for our past accounting section, which is mostly bone samples, bone and teeth, but occasionally may get hairs or stamps and envelopes that have been licked or fingernails once in a while. These samples are much lower quality. So historically, we've typically used Sanger sequencing of HV1 and HV2 of the mitogenome. But with certain past accounting samples, we are still seeing failures with the Sanger sequencing method, only about 6% success rate with some particular samples. And so we needed a new methodology to help improve our results for these failing samples. And I do want to mention quickly that mitochondrial DNA is maternally inherited, so it is not always individual to a person. And that's how we can use it for family reference samples. The reason we're able to use it to identify people in our casework is because we're typically dealing with closed populations and we have military records. So if there's a particular ship that went down, we know who is meant to be on that ship and have records for that. And that allows us to use mitochondrial DNA to identify service members. I do also want to mention forensic protocols validations. I'm not sure how many people are familiar with the forensic community and the standards and the accreditation that is involved in that. But implementing any new protocol into a forensic lab requires a full validation. And that includes some specific studies, including sensitivity studies, precision studies, mixtures, contamination assessment, non-probative case type samples, repeatability, reproducibility. There is a whole document that talks about the type of samples that have to be tested and the ways that they need to be tested. And so then there's also additional work to train up new staff and to implement people into a proficiency testing cycle. So all this is to say that it's labor intensive and expensive to bring novel protocols online in a forensic lab. So forensic labs can't be nearly as agile as the research and academic communities. So with that being said, this is kind of an overview of our validated NGS protocol for degraded bone samples. There are a number of steps. The green arrows are silica column purification steps. The orange arrows are SRSLY bead purification steps. We've got teal and purple arrows that show QC and or quantification steps throughout the protocol. I'll mention that we do include a DNA repair step, which I'll talk more about a little bit later. The library prep is a double-stranded DNA prep kit. And then we are using hybridization capture enrichment instead of PCR enrichment. Our samples are so low quality, there's so little starting material that is our actual target that we just library prep our whole sample, amplify it, and then we do our target enrichment via hybridization capture with a custom bait set that's targeting the full human mitogenome. And then we amplify that captured DNA again. And our sequencing is done on an Illumina platform. So if we look back at our samples, we actually ended up having to validate two different NGS protocols, one for the degraded samples, which I showed in the previous slide, and another one that uses long-range amplification PCR enrichment for our reference quality samples to compare them to. Once we got both of these protocols online in 2016, we've processed about 3,000 case samples, and that has resulted in over 200 IDs. Which is great. We had a 6% success rate with a lot of samples with our Sanger protocol, and now with NGS Online, we're seeing a 60% success rate. But we are still seeing 40% of our samples fail. So we wanted to look into why and find a protocol that could help with our success rate with even those failing samples. One of the groups of samples that seems to be our most challenging is known by a few different names, but they're Korean War unknowns. These samples, of course, age and environmental impact can result in damaged DNA. It can introduce inhibitors. It can introduce exogenous DNA. But with these particular samples, they have undergone additional chemical treatment that has further exacerbated our struggles with getting DNA out of these samples. So some of these were treated with insecticides and deodorants, and then they were transferred to a mortuary in Kokura, Japan, where they were then soaked in vats of 50% formaldehyde for 3 to 5 days. Some were then treated with a fungicide and or covered with a powdered hardening compound. So all of these things together mean that our DNA is heavily cross-linked and it's degraded. There's cytosine deamination. We have a lot of different types of damage going on. So many of these remains from these Korean War unknown remains went through this preservation process and are now buried in the National Memorial Cemetery of the Pacific, also known as the Punch Bowl. And so I'll refer to these samples as Punch Bowl samples or Korean War unknowns, chemically treated samples. We have a number of ways that we refer to these, where they're talking about these samples that underwent chemical treatment that is severely impacting the DNA. So once we started processing samples through our NGS protocol, we got a better idea of their quality. And this image is kind of showing some of our early assessments of the types of samples that we're going through our NGS protocols. So we're seeing mitochondrial DNA read counts on the y-axis and DNA fragment lengths and base pairs on the x-axis. The Kokura, NCMP unknowns, and chemically treated that are outlined in gold, those are those chemically treated Korean War remains that I was talking about in the last slide. And so if we're looking at the low quality and chemically treated samples, these are heavily fragmented and there's not much DNA present. So it's a quality and a quantity issue with these samples. We're seeing mode fragment lengths somewhere between 45 and 70 base pairs. And then if we look at the pie charts along the right, the gray sections are showing the percent that's mapping to our mitochondrial DNA target. And of course, as the quality decreases, so does the amount that's mapping on target. And with the chemically treated samples, I know it's very small, only about 3% on average is mapping to our target. And that means for many of those samples, less than 1% of the DNA is actually mapping to our target. So with these low quality and chemically treated samples still failing, we need to retain as many small fragments as possible in a new protocol. We need increased sensitivity. We need incredibly sensitive kit, and we need a high convergence efficiency for adapter ligated molecules so that we can have enough endogenous content when we get to our hybridization capture enrichment to have a successful capture, and we're not wasting so many sequencing reads on bacterial and other off-target DNA. I do want to mention with a forensic context, there are complications with our mitochondrial analysis of these low quality and quantity samples. And that represents a large portion of our samples. And so some of the things that we'll run into are new mites, despite the fact that they're low quality and quantity. We see new mites in some of our samples. We'll see cytosine deamination, despite the fact that we include a repair step. And also identifying when mixtures are present is incredibly important in a forensic setting. And so we have to, in fact, we don't report any mixed data. We don't interpret mixed data. If we identify it that way, then we reprocess samples until we get a single source profile. And so all of this means that we have very particular, we have to come up with very specific interpretation guidelines and thresholds and standards in order to analyze our DNA, because we're comparing it to other low quality samples and we're comparing it to reference quality samples. So this is all factoring into what we need in a new kit. So where do we look for methods that we can tailor to our failing samples? The answer has been the ancient DNA community. So we started a collaboration with the Max Planck Institute, EVA, and the Department of Evolutionary Genetics specifically. And we used AFDIL samples so we could compare different extraction protocols, as well as 3 library preparation methods. And all of these went through a hybridization, capture, enrichment. We published this data earlier this year in Genes. And so the rest of this talk is going to focus on the results of that collaboration in that project. This is an overview of our methodology. We started with 15 AFDIL samples. Those were 10 from World War II and 5 from the Korean War. And these were all bone samples that ranged in which skeletal element we were using. But they spanned a range that was typical for our casework processing. And so those all went through extraction. We compared some different extraction methods, and I'll talk more about those in the next slide. We also used one of those extraction methods to compare different library DNA repair kits. And then the best extraction method was used to compare the 3 different library preparation kits, one double-stranded kit, and both the MPI and SRSLY Pig Out single-stranded kit. Prior to this project, we had tested some different polymerases for library PCR with SRSLY, and we ended up going with a uracil tolerant polymerase that gave us the most even coverage across the genome. We did do whole genome sequencing of these libraries for some comparative purposes prior to any enrichment. And then at the MPI, they did hybridization, capture, enrichment on all of our samples. This was targeting the human mitogenome, and their method uses 2 rounds of enrichment where the number of PCR cycles are tailored to the input amount so that you're not over amplifying samples. These captured libraries were then sequenced as well, and all sequencing was done on an Illumina platform. And then the data was analyzed with 2 different analysis pipelines. MPI analyzed it with their sediment pipeline that was from an article they published in Nature last year. And then we also analyzed 2 forensic standards in the QIAGEN CLC Genomics Workbench with the Acme plugin. That's what we use at AFDIL to analyze our mito data. So the extraction methods, we ended up looking at 6 different conditions. The green ones there are all a forensic method, our AFDIL method. Those include a demineralization buffer that results in total bone demineralization. And then those samples are further extracted with a buffer exchange organic extraction method. And we used that method to compare 3 different repair conditions: no repair at all, an FFPE repair, and user treatment. And then we did 2 different ancient DNA protocols. These are the Dabney protocol. The 37 degrees is the published incubation temperature, but at AFDIL, we also typically incubate at 56 degrees to better release some of our DNA samples out of bone. And so we took the pellet that remained from 37 degree incubation and re-extracted that with a 56 degree incubation as well. And then we also included an AFDIL Dabney hybrid protocol that was using the AFDIL demineralization buffer for total demin, and then purifying that with the Dabney method. DNA repair, I wanted to mention because I think interest in repair is a difference between the forensic and ancient approaches. Deamination rates are used in the ancient community, obviously, to authenticate ancient DNA. So in that situation, you are probably unlikely to want to repair your DNA and get rid of those telltale deamination patterns. But in a forensic setting, we need to fully interpret our data because, like I mentioned, we are comparing it to reference samples and even other casework samples to reassociate remains. So a DNA repair is helpful in clarifying some of our setting of thresholds and analysis of DNA. And so I wanted to mention these 2 kits quickly. User treatment leaves a basic sites where uracils are present, and then an in-repair follows up to fill in and fully repair those sites. Then there's an FFPE repair kit, which not only repairs deamination, but also nicks and gaps, oxidized bases, and blocked 3' ends. And I will mention that both of these are followed by a silica column purification, and user treatment is what is currently validated in our casework protocol. The first thing we wanted to compare was the extraction methods, and we did this using whole genome sequencing data before any enrichment. The metric I'm showing here is relative informative sequences. So this is published back in 2017 and was also used in our 2022 paper. This was developed at the NPI EVA. And what the informative sequences do is they're a way to determine the proportion of your total sequence DNA molecules that are actually informative, that map to your reference, and then it applies it to the number of molecules in the library prior to any PCR, and that is using qPCR data, real-time PCR. And this is good for comparisons within a sample, but if you're comparing across samples, between samples, it's helpful to use the relative informative sequences where you create a ratio relative to the best method. And so that's what we're showing here is the relative informative sequences. And when we look at this figure, on the left, we see that the AFDIL Dabney hybrid method was very poor performing. It was significantly worse than any of the other methods. The best performing method was the ancient method with a 56-degree incubation. And this method was significantly better than the two AFDIL methods that included a repair. We're losing DNA with the repair and probably primarily with the purification step after the repair. So this was not entirely surprising. But there was no significant difference in informative sequences between the two ancient methods and the AFDIL method that did not include a repair step. They were all comparable. But we thought, what if we had only extracted with a 56-degree incubation instead of doing the 37-degree and then re-extracting the pellet at 56 degrees? And so we decided to simulate what we might recover if all the DNA from both of those extractions was in a single extraction. And when we did that, we see that a merged scenario here would result in a significantly better performance of this ancient method compared to any of the forensic methods that we included. I also want to touch briefly on the different library prep methods, which I'm sure people are very familiar with. But on the left, we have the NPI single-stranded DNA 2.0 method. This was published, there was an update to it in 2017 and then another in 2020. And the 2020 method is their fully automated method, which is what was used in this project. But just very briefly, that method uses biotinylated adapters. So library prep is completely performed on bead. This includes wash steps as opposed to using a SRSLY or silica column purification. Adapters are added in different steps, and so full length adapters are present after completion of library prep. And the SRSLY method, on the other hand, does adapter ligation of both adapters in a single step, and blockers are used to ensure that these are ligated in the proper orientation. Both of these library prep methods use a type of splinted adapter, and library PCR can be used to add barcodes to the samples to differentiate them bioinformatically. I do want to mention quickly that when we started looking at data, we saw some difference in coverage levels of very specific small regions of the mitogenome. And these are differences between the SRSLY and NPI libraries when we were looking at them with our CLC pipeline at AFDIL, but the NPI, when they were analyzing it with their analysis pipeline, they were seeing no differences. Everything looked the same to them. And so when we were looking down, we were in the weeds at this point. We were using a super sensitive method. We're looking at our negative controls and our worst quality samples, and we, comparing to older validation data, realized that we were seeing small bits of pig DNA that was mapping to the human mitogenome. And so we realized that the Max Planck used their sediment pipeline, which is mapping or is pulling out sequences that map to various references. So you're only seeing what's mapping to your target. And at the AFDIL, we were just mapping straight to our human reference. So what we did was create a new workflow called Pig Out. And this did a sequential mapping where it was mapping with high stringency to a pig reference first, and then mapping everything that was unmapped to the human reference. And so everything downstream of this was using that workflow. And I'll mention that the source of this has been identified from a third-party buffer, and that's been replaced. And honestly, we were using so many PCR cycles, we were so down in the weeds with these samples that not likely to be seen in many situations. But I did want to show this. These are read mappings, both before and after that pig out workflow. And you can see the arrows indicating these very specific regions where you had much higher coverage than in other regions. And when we used the pig out workflow, we saw negligible reads mapping. And this is consistent with our clean controls with any of our other methods. So this workflow was effective in removing the pig DNA, but it also didn't result in serious loss in coverage, even in these regions of the mitogenome. So when we started to compare the library prep kits, we again used relative informative sequences and WGS data for this. What we found was that the double-stranded DNA method was significantly worse than the single-stranded prep methods. Again, this is not surprising. We knew we had very degraded DNA and that we needed a more sensitive method. And we did find that the MPI and SRSLY methods were not significantly different in their informative sequences, relative informative sequence levels, slight bit more variability with the MPI method here. We then wanted to look at the percent of sequences mapping to the human genome. And I'll go ahead and mention, in the next few slides, I'm going to show some similar figures. So to orient you to these figures, along the x-axis are individual samples. So each bar is a sample. The purple bars are AFDIL samples included in this project, and the green bars are actually previously published ancient DNA samples. These are ancient samples from late Pleistocene modern human skeletal remains, and they range from about 37,000 to 46,000 years old. And so we were able to compare our AFDIL samples to this previously published data. And then on the y-axis here, we're seeing the percent of sequences that are greater than 34 base pairs. We did trim, so it was more consistent with the ancient published data. And the percent mapping to the human genome, that is our reference. And so what we found is that the AFDIL samples varied. Some of them had a high percent map, but many of them were pretty low percent mapping to the human genome. And so when we look at the main left part of the figure that's kind of zoomed in on the lower samples, the solid black bar is the average for the ancient samples, and the dashed bar, the dashed line is +2 standard deviations. And we do see that some of our samples are falling in this range that is consistent with the ancient samples. But we do see more variability in our samples than with the ancient samples. We also looked at average fragment size compared to these ancient samples. And we see a much consistency between not only within our AFDIL samples, but also between AFDIL and ancient samples. This is consistent with what we expected and what we found before, which was these samples are highly fragmented, many of them. So we're seeing a range of 44 to 64 base pairs, and with a mean of about 49 base pairs. We also wanted to look at deamination rates. So we use C to T substitution to kind of assess the deamination rate. And what we found here, if you look at the figure on the left, the 10% indicates about what MPI EVA uses and other ancient labs use potentially to authenticate ancient DNA. Anything at or above this level would not be considered a modern contaminant. And so again, the black bars is the average for the ancient samples, and the dashed lines are ±2 standard deviations. And we are seeing that most of these AFDIL samples are meeting this 10% or exceeding this 10% deamination threshold. We saw a range of 8.8 to 30.7%, and then a mean of about 18.3. So in fact, we are seeing damage patterns and rates that are consistent with ancient samples, but not quite as high as many of these 40,000-year-old ancient samples. And then on the right, that's the pattern of damage from the terminal 5' end to interior to the DNA fragment. And we see the pattern that you'd expect for ancient DNA, which is higher deamination rates on the N and in about 3 bases with much lower rates towards the center of the fragments. So our most challenging AFDIL samples are comparable to ancient samples in many ways. So we're looking to the right community to improve our methods. After hybridization, capture, enrichment, we wanted to see what percent we were seeing mapping to our target genome, and where the mean was 7.88% prior to enrichment. After the hybridization capture, we saw 60 to 94% mapping with the MPI method and 60 to 84% mapping with SRSLY. So we were seeing a significant improvement in our on-target percentage an order of magnitude effectively. So that very fine-tuned capture protocol that they use is very effective in enriching for our target. The next thing we looked at was average coverage. This was average coverage across the whole genome. And this is a metric that is useful in forensic context as a way to triage samples and decide what data is reliable and what data would be considered inconclusive based on validation results. So we definitely wanted to see average coverage of each of these samples. Again, on the left side, we see the MPI libraries, and on the right side, we have the SRSLY libraries with coverage on the y-axis and the same set of samples reproduced in with each library prep method. And we're seeing very comparable results here. With the MPI kit, there was a range of 6x to over 300x. And similarly with the SRSLY kit, we were seeing a range of 7.3x to over 300x. The colors of the bars in this figure is representative of our initial success rate with our casework method, our double-stranded validated casework method. So the red bars failed our casework processing and were not reportable. The green bars were relatively high coverage, over 100x coverage, and our low coverage ones are in blue there. So what was really incredible here was that these samples that failed on casework are now generating usable data with both of these single-stranded library prep kits, where we were not getting usable data before. And just to point out the lowest quality samples, this is one of the low quality samples, 1487. And this one we see slightly higher coverage, average coverage with SRSLY. And then this other sample is one of our other next lowest quality one. And the opposite is true there. So really comparable results between these two library prep methods. In addition to the average coverage, we wanted to see our breadth of coverage, our consistency across the mitogenome, because that is also important in how much of our sample that we can report out, what ranges we can report out. So this is the percent of the human genome covered at a 5x level. And what we're seeing here is, again, very comparable results between these two library preparation kits. These are the two samples boxed on the bottom that were those low quality ones from the previous slide. And as they're the lowest quality, we are seeing a reduced coverage. We're not seeing, full mitogenome coverage, but these failed completely in casework. So this recovery is really a huge improvement for us. In addition, the actual variant calling is incredibly important in forensics, not just identifying a species that's present or verifying the age of the DNA. We have to compare to reference samples and individualized samples compared to other casework samples. So variant calling concordance is something that we needed to consider, and we did this using our CLC pipeline that's used at AFDIL. And what we're seeing here is a high level of concordance between the two methods. When we're calling at such a low level, we are seeing even minor differences in coverage or percentages of minor variances resulting in slightly different variant calls. But the things that we're seeing here are not concerning discordances, because it's so much due to the low coverage plus damage plus small percentage differences. And at AFDIL, we process every casework sample twice. It goes through this whole process. It would go through this whole process twice. So any stochastic errors going on at these very low levels would be mitigated by reproducing the data. And then if data is single source, we will merge the sets of data for an overall profile calling. So we also saw high complete consistency in haplogroup prediction with these two methods. So some quick analysis considerations with these low quality and quantity samples. As I was saying, these are incredibly sensitive kits, these single-stranded library prep kits. So when you're looking down at such a low level, it is worth looking carefully at negative controls. We always include negative and positive controls in forensic processing. So those are very helpful. We also, it was incredibly helpful to have two different analysis pipelines we were using to analyze this data. That helped us in investigating things that we wanted to look into and really digging into the data, as opposed to just having a single analysis pipeline where we may have some limitations on what we can look at. So it's just looking very carefully at that data, doing contamination assessments, doing low level, looking at differences between different ranges and across samples and between samples really helped us out in the situation. And it is interesting to consider using a pipeline that uses multiple reference sequences that you're mapping to, as the NPI uses with their sediment pipeline. And I will mention we're currently validating, as I said before, a SNP assay. And all of these considerations are really helpful in us thinking about how to analyze that SNP data, because we're calling at incredibly low levels for that as well with our highly degraded samples. So in summary, some AFDIL samples exhibit traits that are consistent with ancient DNA samples. If the context was completely unknown, they might be considered ancient samples. And the DABNI extraction developed by the ancient community by MPI improved our recovery of small fragments as it was intended. And while a DNA repair step may ease analysis in a forensic setting, we do lose DNA from the repair, but also certainly from the purification that follows it. And this is a problem for our most challenging samples. So it would be helpful to be able to bioinformatically manage some of the issues with the DNA damage for our most challenging samples, so that we're not losing our fragments that we need in order to get reportable data. And then the single-stranded DNA library prep did improve our results from samples considered low quality or failed with the DNA double-stranded DNA prep kit, and the MPI and CSV methods were quite comparable and both worked incredibly well. And I will say that the sensitive and fine-tuned hybridization capture enrichment protocol that they developed at the MPI was important for us getting enough of our target, adequate recovery of our target to get these reportable results. So with that, I would like to acknowledge the rest of the folks and the ETS group at AFDIL. It's a collaborative small section, and they've all been involved in various projects. I would like to thank the MPI, Elena specifically. She was the co-first author on the paper and did an incredible amount of the work for that, as well as Charlotte Marshall. And then a thanks to Dr. McMahon and Colonel Briones for allowing me to present this work, SNA International as well, and Claire for allowing me to present today. And my contact information is listed there on the right side, as well as Elena's, since there are certain parts of the project that she took point on and others that we took point on. So with that, I can answer any questions if there are some.
Thank you so much for the presentation, Tyler. I believe we have a couple of questions. I'll read them out and then you can address them. We have a question from Kelly Harkins, who is the CEO of Claire Bio. Can you describe again the details of your combo of the 37 and 56 DABNI extraction methods?
Yes. So we took a bone powder aliquot of about 0.2 grams, and we added the mill of DABNI buffer, incubated it overnight at 37 degrees, and then used the high pure viral spin column from Roche to purify it. But we retained the pellet from the initial overnight extraction, and we added more DABNI buffer and incubated that overnight at 56 degrees and did the same purification afterwards. So it was just the remaining DNA pellet, bone powder pellet. We basically repeated the extraction just with a higher incubation temperature.
Great. The other question is the MPI library prep available to purchase?
I don't know. That would be a question for Elena or someone else at MPI. I know they've published it a lot, so a lot of the details are available through various publications, but I'm not sure if it's for sale. But if that person will send me an email, I can ask about that.
And we have another question by L'Oreal. Do you modify the SRSLY protocol in any way? Do you dilute the adapters?
When we were doing this protocol, we did use a uracil tolerant polymerase. We did not dilute the adapters for this protocol, but we didn't dilute the adapters for this protocol. And so but the purification did work pretty well to remove our dimers. And I guess we followed the ancient DNA protocol that preferentially maintains the small fragments, including the modification to the ampure that includes the isopropyl alcohol and tris HCl addition.
I must add to that question. So we now have a SRSLY uracil tolerant kit available. So previously, of course, a lot of the ancient DNA applications, we would ask them to replace it with a uracil tolerant polymerase, but now we have it as a kit option.
Yeah, I did see that. That's wonderful.
Yeah. There's another question. I saw you used NEB library prep. Do you think the user step that cuts the adapters can cut at your damaged ancient DNA?
In our casework protocol, we do the damage repair prior to library prep. So at that point, and we use user twice in that process. We use it at the beginning after extraction to repair our DNA. And then the way that the adapters are created in the NEB protocol, they do purposely have uracils in a looped adapter format. And so it's supposed to reduce the dimer formation, but then you use a user treatment to actually create the Y-shaped adapters for downstream processing. So with the NEB kit, you have to do a repair before you start library prep, because if not, yes, it's going to chew your any damaged DNA up. Bases due to uracil deamination, which is why in the collaboration with MPI, where we didn't do repair, we used a Kappa kit, because that doesn't use the same user treatment to kind of create the Y-shaped adapters. So we didn't have to do library or DNA repair prior to library prep with the Kappa hyperprep kit.
I had a question. So with the mitochondrial DNA that you extract, do you see that the mitochondrial DNA sizes are different from the overall DNA sizes, or it's the same for all of them? And do you see a preferential performance between the kids based on sizes?
Right. So I think we typically go for mito, because since it's circular and since there are more copies than cells, there's just more of it there over time. So I think we just see more of a range in fragment size with mitochondrial DNA, I'm thinking, just because there's more there. So there might be enough that's of sufficient length to work with protocols, whereas with with traditional forensic protocols that use an amplicon, like a multiplex amplicon strategy, to generate profiles, those fail with our low quality samples. And even the Sanger sequencing, if we're targeting 80 base pair to 150 base pair fragments, a lot of our samples, our worst quality samples, are still failing with that as well, which is why we needed to adapt NGS protocols. But so in order to use nuclear DNA, we're having to pivot to SNPs with an NPS protocol, because then that's amenable to our small fragments. But so I don't know if the mitochondrial DNA is necessarily like less degraded, but there's just more of it, I think, that's there. So we just have more fragments of varying sizes available to us for processing.
I had a follow-up question on that. So because mitochondrial DNA is protected in an organelle, do you think there is fewer deamination and things like that? Is there like a comparative study that people have done to look at this?
Oh, I don't know. That's an interesting question. I'm not sure, because we were looking at WGS data. So the deamination rates that I was showing in this presentation were based on the nuclear DNA, but I don't think I didn't compare that directly to results from the captured data. So that's an interesting question. I'll have to look at that.
I think L'Oreal has another question. Which spry beads did you use?
We used Ampure.
Okay. If there are no more questions, please join me in thanking Tyler today. As she mentioned, you can write to her with additional questions that you have. If you have seriously specific questions, feel free to write to us. We will, as attendees, we will share this talk with you, and in that email, we'll share the contact information for Tyler and for Claret. With that, I'd like to thank you. I think Kelly is also saying thank you. She's been super interesting. Of course, it is so fascinating. And as I mentioned to you before, I come from a clinical background, so forensics and ancient, I nerd out on this completely. So thank you again, Tyler, for a fantastic talk.
Yeah, thank you. Thank you for hosting me.
And thank you to all the attendees for setting aside time for this.